Hello. The first thing I want to do this week is clarify some information from last week when we were talking about genome-wide association studies and polygenic risk scores. Last week, we looked at a number of Manhattan plots. These are plots where along the x-axis you have chromosomal position. Here I've just made up 6 different chromosomes. And on the y-axis, you have the significance of the association of a particular SNP with your phenotype of interest. When you do a genome-wide association study, one objective might be to find a particular locus that is significantly associated with your trait. That is, an individual locus that crosses some genome-wide significance threshold. In the example I've drawn here, there are no individual dots that are above this genome-wide significance threshold. We saw this happen in the paper, that coral paper that we discussed too. And the authors of that paper said that this might have been due to the fact that their study was underpowered. That is, they didn't have enough individuals in their study. They did not get enough genetic and phenotypic information from enough individuals to, to have a chance of finding a significant association. That's because the trait that they were studying, they said, was polygenic, or it was determined by lots and lots of genes. And that's statistically hard to find. It means that instead of just one particular genomic locus that is in charge of the trait, like Mendel's peas that were round or wrinkled just because of that R locus, instead they might be looking for hundreds of different places along the genome that have subtle contributions to the trait. So it is statistically difficult to find a single genomic locus that is significantly associated with the trait. But there's one thing that the authors of this study did do with their data, and that anyone can do with their GWAS data, even if you don't find a individual locus that is significantly associated with your trait. Here is an example of what your data from a GWAS study might look like in table form, so not in the Manhattan plot, where each dot in the Manhattan plot was a locus. Here, instead, I have a row in this table for each locus. So I've given the locus a name, let's say locus 1. Locus 1 is a variant that we discovered in our population. So now I'm going to say, what were the 2 alleles that I found to be variable in the population? And just for ease of this example, I'm going to call the first one that we see here the effective allele, and then I'm going to call the other one just the other allele. Now, the effective allele is the one that's going to be positively associated with our trait of interest. Let's say risk of heart disease in humans, just to keep things straight. So I have the effective allele, that is which allele is associated with an increased risk in our population, whatever the other allele was that I found in the population, some p-value, and an odds ratio. You might want to go back a couple weeks to the video where we talked about odds ratio to remind yourself that this is a measure of the strength of the association. So I only put this p-value column in here right now just as a demonstration that I can do this analysis that I'm about to show you for all SNPs. So I get these data from a GWAS study, but now I'm not interested in finding an individual locus that passes some significance threshold. I can still use data from all loci, even if I don't see a statistically significant association with my trait. So what can I do with this data? In particular, I'm going to use this odds ratio, which is the strength of the association, or kind of you can think of it as the weighted risk that this allele at this locus confers on the phenotype. And now let's say I have genotype information for 3 individuals at these 3 loci. So I have individual A, B, and C. Individual A is, let's say, homozygous AA at locus 1, homozygous TT at locus 2, and homozygous CC at locus 3. Let's say that individual B, we genotype them. We find that they are heterozygous at every position. And individual C, we genotype them and find that they are homozygous for the other allele at every position. So TT, GG, and GG. And remember, none of these loci are significantly associated with the trait of interest. So it's not like knowing the genotype at any one position is going to give me useful information about whether or not this person is likely to have our trait of interest, or in our example that we've been using, be at high risk of cardiovascular disease. However, we can still get information by summing up all of the effects of the loci that we see. And that is what we call a polygenic risk score, or where's my pen? PRS. And I'm going to show you how you could calculate a polygenic risk score for each individual, which I will call J. So PRS sub J is what we are calculating. And we calculate this by summing, using that sigma sign to indicate that I'm going to be summing for every locus, which I will indicate with the letter I. I'm going to sum up all of the effects of the loci in individual J to calculate their polygenic risk score. So what am I summing? Well, the first thing that I'm summing is the effect size of that allele. The effect size, that's that odds ratio, or how strong is the association of that locus with that trait. And I'm going to be multiplying that by the genetic information I have for each individual, or in other words, how many copies of the allele, of the effective allele at that locus, does that individual have? So let's go through one example and then maybe you can do the rest of your own. So right now I'm just going to focus on individual A, so calculating the polygenic risk score for this individual. So for locus 1, I know that the effect size, or the odds ratio of locus 1, is 2. And I also know that the G, or the number of effective alleles at that locus in my individual at this locus, is 2, because this person is homozygous AA, and A is the effective allele that has that risk of 2. I can keep going down the line here and calculate that same value for the second locus. Now, the effect size of the effective allele here is 1, that's the odds ratio. And how many copies of the effective allele does individual A have? Well, they have 2. They're homozygous T. I can do the same for locus 3. The effect size of the effective allele is 3. I multiply that by the number of copies of the effective allele this person has, which is 2. And now, as the equation states, I sum up all of those values. So this is a weighted sum, weighted to the strength of the association and the number of copies of that allele that each person has. So for individual A, I get a value of 12. You can pause the video here and fill in the rest if you want to check your math. I filled in the polygenic risk scores for individuals B and C. Notice that this first value in each component never changed because the odds ratio, or the strength of the association of that effective allele with the phenotype, is a constant value. That's what I discovered from my genome-wide association studies. So that isn't going to change from person to person. What changes is the number of alleles of the effective allele that each individual has. So our heterozygous person, or the person who is heterozygous at each of these locus, they're all multiplied by 1. And the individual who was homozygous for the other allele, that individual is 0, which obviously results in a polygenic risk score of 0 for this individual. So what you can see here numerically, which you might have seen with your eyes before, is that individual A has the highest predictive risk according to their genotype across these 3 loci, and individual C has the lowest predicted risk. And just to be clear, that you know, these are all very simplified, made-up numbers. Your actual data would look really different. Your odds ratio wouldn't actually be presented like this in most cases. These are just very simple numbers made up to help our example here. And obviously, you would have many more loci for your different individuals. There are also different formulas that you can use to calculate your polygenic risk score. And there are different ways that you could choose the number of loci that you sum up in order to calculate that score. And lastly, one thing I want to point out here is that this formula, the way that we have it set up right now, assumes an additive model of the alleles. It assumes that having 2 copies of A is twice as harmful as having 1 copy. And you might remember some things we talked about way earlier in the semester about dominance and recessiveness. Sometimes there is an interaction between alleles that doesn't make the combination of their effects purely additive. So, for example, some phenotype may be conferred by the homozygous big A that is not conferred at all in the heterozygote. So, just to be clear, some caveats to this exact math that we're doing in this example. And lastly, I would like to remind everyone that a polygenic risk score is a useful summary, but it is not predictive of the fate of any one particular person. So any one particular person could have a low poly risk score and let's say low cholesterol. In our example, I just made a red dot there. There may be an individual who has a high polygenic risk score and also has high cholesterol. But like we saw in class, when you actually look at all of the values in a population, you'll see huge variance in all of the traits and all of the polygenic risks. There may be an overall trend in the population that can be informative for future studies, but that polygenic risk score is not a fate of any one particular person.